Moving on to some research that I find exciting, which is looking at omega-3 consumption and supplementation in animals and the effects on lifespan and the effects on disease. So this is a situation where we can test very clearly what happens throughout the entire lifespan of an animal with an intervention like this. We can't do this with humans. You can't have a human that essentially from birth is given omega-3s and then you see what happens 80 years later with this huge cohort. But you can do this very easily in animals, and you can see the effects on lifespan. You can see the effects on different disease processes, and we'll dig into the research showing it. It's exciting stuff.
Just a caveat before you start, Jay, there is a cohort of people that is currently taking omega-3s from birth all the way until perhaps their entire lifespan currently. I just want to say, between formula feeding and then supplemental omega-3s for children in their milk while they grow up, the DHEA fortified milk and whatnot, that is currently happening despite the inconclusive nature of the research.
That's true. And despite the, research, the conflicting research specifically on infants and infant outcomes and things like that. The other thing worth mentioning too, I know you're saying this semi-facetiously, of course, there's not like a trial there, but we will be looking at populations that have very high omega-3 intake. And this is not in the context of formula feeding and adding supplements to food, but rather getting their omega-3s directly from fully natural whole foods like fish and whales, blubber and things like that, seals, being the Inuits and Eskimos. And we'll come to that later. And it actually doesn't support that even consuming the omega-3s from the right sources in large amounts is a good idea. So, but anyway, coming back to this animal research, we'll go through a handful of studies here. The first one is titled Dietary Supplementation with Lovasa and Krill Oil Shortens the Lifespan of Long-Lived F1 Mice. And Lovasa, for reference, is a pharmaceutical grade fish oil. So, top quality here. And then also krill oil as well, which is sometimes suggested over fish oil because it has some antioxidant capacity with the astaxanthin. Yeah, exactly. So they state here, they conclude that marine oils rich in omega-3 polyunsaturated fatty acids have been recommended as a preventative treatment for patients at risk for cardiovascular disease or diseases. These oils are the third most consumed dietary supplement in the USA. However, evidence for their health benefits is equivocal. Individually, Lovasa and krill oil non-significantly shortened median lifespan by 9.8 and 4.7% respectively. Lovasa increased the number of enlarged seminal vesicles 7.1-fold. Lovasa and krill oil significantly increased lung tumors 4.1 and 8.2-fold, and hemorrhagic diathesis 3.9 and 3.1-fold. Taken together, the results do not support the idea that the consumption of isolated omega-3 fatty acid-rich oils will increase the lifespan or health of initially healthy individuals. So, yeah, I don't know what more to add. We're seeing increased, various diseases, potentially reduced fertility or male fertility with those enlarged seminal vesicles. We're seeing a non-significant, but a large effect size. It was non-significant, but large effect size as far as a shortened median lifespan of 9.8 and 4.7%. So, these are some pretty clear findings, and we'll go through some more clear ones, suggesting that supplementing with pharmaceutical grade fish oil or krill oil for a lifetime is probably not a great idea.
Yeah, and just some minor bleeding and cancer along the way. Nothing
Right.
too serious. Nothing too serious.
Now,
again, I'm making jokes about it, but I want to make this doesn't mean that people are going to necessarily get cancer and bleed out from fish oil, but just basically what you're seeing is if fish oil is so beneficial, you're not seeing the benefit with species of long-lived Formula One driving mice.
Right. Yeah, exactly. Do you want to go through the next one, Mike?
Yep. So the title of this one is Long-Term Intake of Fish Oil Increases Oxidative Stress and Decreases Lifespan in Senescence Accelerated Mice. So just a caveat before we jump in, or kind of like an explanation before we jump in. The previous set of mice generally lived a long time. And this set of mice has a proclivity to not live a long time. They have senescence, which is basically like the cells move into a state of non-replication and kind of like this like limbo state, which is usually associated with shorter lifespan. So these mice don't live as long. So we're going to see the effects of fish oil in these mice, and the title already kind of gave it away. But
sorry, Mike, I think that it looks like they're more prone to, like, they experience cognitive decline and Alzheimer's type effects younger. I don't know if they actually don't live as long. I think it's more just that they experience those things.
They just have the
sooner.
effects of the chronic diseases. Yeah.
Yeah, it looks like it, so that they can study those sorts of things in a little bit more detail. I'm not 100% sure on that. It doesn't matter too much, but yeah.
Yeah. Either way, you have a set of mice where their cells move into senescence quicker. So basically, like much less metabolically active cells that aren't replicating anymore. And it's usually the senescent cells that are associated with chronic disease states. It's not a good thing. So the researchers start here and they say the SAMP8 mice, so these are the senescence accelerated mice, that's what I assume the SAM stands for, fed fish oil, did not have a longer maximum lifespan and had a shorter average lifespan than mice fed safflower oil. So there we go again. The safflower oil and the corn oil are outperforming. To examine the mechanism underlying these results, the effects on oxidative stress of long-term ingestion of fish oil were examined. SAMP8 mice fed fish oil for 28 weeks showed strong oxidative stress that caused hyperoxidation of membrane phospholipids and a diminished antioxidant defense system due to a decrease in tocopherol compared with mice fed safflower oil. These findings suggest that that intake of fish oil increases oxidative stress, decreases cellular function, and causes organ dysfunction in SAMP or SAMP8 mice, thereby promoting aging and shortening the lifespan of the mice. So, I mean, the oxidative stress here is likely because the fish oil has much more double bonds present in the fats. So we kind of already covered this specific piece, especially compared to the safflower oil, even though the safflower oil should technically increase more of the highly inflammatory mediators, but essentially, and these are mechanisms that we already covered previously, but the increased oxidative or peroxidative stress of these fats inside the membrane so inside the actual structure, the cell membrane of the cells, basically destroyed the antioxidant defense system, depleted vitamin E, tocopherol, and then decreased the lifespan and the cellular function and increased organ dysfunction for these mice. So essentially, the way I kind of view this is if you build your house out of really crappy materials, then the cells or the house will not function very well and they will have a much shorter lifespan. You're going to have to get your roof repaired probably in 5 years instead of 30. And so basically that's kind of what you're seeing with these rats and with when you incorporate these highly unsaturated fats into the tissues, in the form of fish oil.
Yeah, exactly. We're basically seeing the exact mechanisms played out that we were looking at earlier in those studies that were showing increases in peroxidation being associated with shorter lifespan, increased peroxidizability index being associated with shorter lifespan across species. And they had then tested it. They gave those rats fish oil, saw increased oxidative stress, and said, well, probably is going to decrease lifespan. And here we see exactly that. And again, we're not comparing fish oil to olive oil, to coconut oil, to something that's very stable. We're comparing it to safflower oil, which is very high in omega-6s, also very prone to peroxidation, but just not quite as prone as fish oil. And you see hyperoxidation of membrane phospholipids, diminished antioxidant systems, as well as shorter average lifespan compared to safflower oil. So pretty clear demonstration here of exactly what we would anticipate to be the effects of fish oil.
Yeah, and it follows the trends that we kind of already discussed with peroxidizability index previously, where the more double bonds, the more unsaturated the fats are, the more peroxidative stress you induce, and then the more problems you essentially have, especially within the cell membranes and the cellular structure. The other thing I want to point out here, just slightly tangentially, but a lot of people are, there's all these studies that show that fish oil decreases oxidative stress. And what I want to mention here is that fish oil, at least from the mechanisms that I've gone through, is decreasing oxidative stress by creating peroxidized fatty acid mediators and then triggering hormetic pathways, or basically triggering cell defense pathways to clean up the mess. And so the question becomes, okay, maybe inducing the different antioxidant enzymes, glutathione peroxidase, superoxide dismutase, catalase, etc., perhaps that will decrease oxidative stress for a period of time. But it's in doing it because you already have the peroxidation from the beginning. And then the further problem is that sustainable long term? Can you run a marathon continuously? And what I mean by that is, can you put a stressor on the system, on the body, or on the cell over extended period of time, and to get this supposed outcome? And again, and this kind of goes into some of the hormetic arguments that we discussed previously, but I just wanted to put that into perspective there, because it's the antioxidation you're seeing is not because the omega-3s are specifically antioxidants. It's because they're creating lipid peroxidation products that are triggering cell defense, which is a completely different mechanism than increasing your vitamin E or your vitamin C content.
Yeah. And we saw that earlier in one of those studies. I believe it was the one on salmon. But yeah, and we've talked extensively about hormesis. Of course, I'll be linking back to those episodes. But yeah, to say that that's a good thing, I think, is like to say that it's beneficial to, increase antioxidant defenses by causing oxidative stress is not something we would agree with. And there, we go through all the reasoning for that in that series. So I'll definitely link back to that. So moving on to the next paper here. It's titled Excess Omega-3 Fatty Acid Consumption by Mothers During Pregnancy and Lactation Caused Shorter Lifespan and Abnormal ABRs in Old Adult Offspring. So the first quote here, they state the excess offspring, and this is excess meaning excess fatty acid consumption. And then when they say control, it's a normal amount of fatty acid, of omega-3s, and then deficient is deficient in omega-3s. So these are the three groups that they're comparing. So they state the excess omega-3 offspring had shorter lifespans compared to their control and deficient cohorts. And this is 506 days on average for the excess, 601 for the control, and 585 for deficient, which is a difference between the excess and control of 16%, I believe, about 15-16%. So a huge difference in lifespan between those two groups. When the study terminated on postnatal day 640, the excess offspring had a higher incidence of presbycusis than the control and deficient groups. A difference of, so it was 33.3% for the excess, and then the control was 4.3% and deficient was 4.5%. So we're talking about an 8 times difference there, and a persistence of other sensory neurological abnormalities and lower body weights in old adulthood. So that is quite concerning to see the effects of excess amounts of omega-3s of offspring. So again, this is looking at what happens when the parent is fed, the mother is fed excess omega-3s.
So the presbycusis is age-related hearing loss, just for anyone who's doesn't know the term. It's just a medical term.
Yeah, and seeing major effects here. I mean, huge percentage differences in lifespan and also in these sorts of disorders, neurological disorders and things.
It's also interesting that the deficient didn't have such decrease compared to control. Like it was marginally less, at least on a percentage basis, in comparison to the excess versus the control.
Right. Yeah, you're saying in lifespan?
Yes.
Yeah. So 601 days for control, 585 for deficient. So a little bit less, although not sure if I don't think the difference between those two is significant, actually. Not 100% sure. And then looking at the excess, it was 506 days. So a huge difference there. So moving on to the next quote here, they state, post hoc comparisons indicated that the excess offspring weighed significantly less than their control and deficient cohorts, whereas the control and deficient offspring did not differ from each other. So again, now we're seeing just in not birth weight, but the final weight of the offspring. So their general body weight is considerably lower when they have excess amounts of omega-3s, which is a sign of major issues with development. Then they state, in conclusion, omega-3 fatty acid overnutrition or imbalance during pregnancy and lactation had adverse effects on lifespan and sensory neurological function in old adulthood. The adverse outcomes in the excess offspring were likely due to a nutritional toxicity during fetal and or neonatal development that programmed them for lifelong health disorders. The health implication is that consuming or administering large amounts of omega-3 fatty acids during pregnancy and lactation seems inadvisable because of adverse effects on the offspring. I don't know if there's too much that I want to add there. It's so clear based on what they said and incredibly concerning. So I do want to move on to two studies that are not looking at lifespan changes in animals, but instead are looking at different disease processes and other effects that you can see in animals that are fed lifelong high amounts of fish oil versus other fats. And I think this is some evidence that we've talked through a bit earlier, but some very clear and important things to go through. Do you want to share this first one, Mike?
Yeah, I think these come from the fatty liver, some of the fatty liver studies we did, correct?
Yeah, we probably did
episodes.
talk about. Yeah, we definitely talked about both of these during the fatty liver episodes. Yeah.
The first study here is titled Gene Pathways Associated with Mitochondrial Function, Oxidative Stress, and Telomere Length are differentially expressed in the liver of rats fed lifelong on virgin olive, sunflower, or fish oils. What they start out here, they say, this study investigates the effect of lifelong intake of different fat sources rich in monounsaturated, so that's virgin olive oil, N6 polyunsaturated, so that's sunflower oil, or N3 polyunsaturated fish oil fatty acids in the aged liver. Virgin olive oil led to the lowest oxidation in ultrastructural alterations, so that's kind of the structure of the liver of the cells. Sunflower oil induced fibrosis, ultrastructural alterations, and high oxidation. Fish oil intensified oxidation associated with age, lowered electron transport chain activity, and enhanced the relative telomere length. According to the results, virgin olive oil might be considered the dietary fat source that best preserves the liver during the aging process. The lipid profile was analyzed in the liver mitochondrial membrane, since we have previously demonstrated that this is a good marker of dietary fat intervention at different tissue levels, including liver, brain, heart, and skeletal muscle. The lipid profile resembled that of the original composition of oils used in the diet, with animals fed on virgin olive oil showing the highest percentage on oleic acid, those fed on sunflower oil having the highest percentage of linoleic acid, and animals fed on fish oil registering the highest percentage in docosahexaenoic acid. So the linoleic acid is omega-6, and the docosahexaenoic acid is omega-3, and an oleic acid is a monounsaturated fatty acid. So olive oil is very high in the oleic acid, sunflower oil is very high in linoleic acid, and then fish oil is obviously the fat that's very high in docosahexaenoic acid, otherwise known as DHA. And basically the liver or the liver mitochondrial membranes reflected the actual fatty acid composition within. Obviously, we talked about there's a particular range, but you could tell which mouse had which type of fat when looking at the liver mitochondrial membranes because of the change in percentage of these major fatty acids. And essentially what you're seeing, what is the outcome of these changes, is the virgin olive oil, the rats fed virgin olive oil, they actually did pretty well overall. They didn't really have a high amount of oxidative stress or oxidative damage, and the actual structure of the cells of the mitochondria of the liver were fine. Whereas the fish that had the sunflower oil, they had fibrosis. So fibrosis is usually damage to the cellular structure and then a replacement with just collagenous tissue, or basically inactive tissue that doesn't really do anything. It's usually indicative of a damage. And then they had high levels of oxidation, and then they had the ultrastructural alterations as well. So the cellular structure was not really what it should be. It was a bit damaged. And then the fish oil had probably the worst oxidation, at least implied by what they're saying here. And then interestingly enough is the electron transport chain activity, the actual flow of electrons in the mitochondria to produce ATP was decreased in the fish oil specifically. They note this here, which is very interesting overall. But it also makes sense with the mechanisms that we've discussed previously. We're incorporating fish oil into the membrane actually dissipates the proton gradient and then leads to issues with production in ATP. And then also makes the membranes very susceptible to oxidative stress, which you're also seeing here overall. So basically, I guess if you want to think about this very simply, olive oil perform the best, safflower oil or sunflower oil perform the second best, and then fish oil perform the worst of these three fatty acids. And basically what you're seeing is a monounsaturated versus an omega-6 polyunsaturated versus an omega-3 polyunsaturated fatty acid.
Yeah. Yeah. And it's demonstrating very clearly a lot of the mechanisms that we had discussed earlier: increased oxidation and the extreme disruption in electron transport chain activity. There was also a major decrease in mitochondrial density in the fish oil group. So yeah, definitely not outcomes that we're looking for in any intervention. And yeah, pretty clear to see here when you're looking at lifelong consumption of fish oil in these animals.
Yeah, and again, it's not to say that this is exactly what happens in humans, because there's a degree of difference. Although the cellular structures are indeed relatively similar to some extent between humans and mice, but the overall picture here is maybe you have this benefit of fish oil not fish oil. You have this benefit of omega-3s with the RBC phospholipids. And so you're thinking, oh, I'm going to go take fish oil. But when you start to see information like this is a huge pause, at least for me, because you're seeing such a striking negative effect that's in line with some of the other cellular components. And then you're seeing this in animals where you can actually test the effects across the lifespan, and we can't test the effects across the lifespan for humans. So we won't be able to see some of these things play out over the long term in humans, at least in studies. So yeah, this gives that context for me.
Right. And even though we didn't see it necessarily lifelong in humans, we did reference quite a few studies showing massive increases in lipid peroxidation, oxidative stress, issues with mitochondrial respiration in humans when omega-3 supplementation was used as an intervention. So we're seeing the same things here. This is not conflicting at all with what we were seeing as far as those effects.
Yeah. So it doesn't conflict with the mechanisms. It doesn't conflict with those effects. You just can't really see the lifespan changes directly. It's kind of hard to parse those out specifically.
Right. Yeah, definitely. And there's one more study I want to go through here, again, looking at some effects on the liver as a result of fish oil. And this was also one that we went over in that fatty liver disease series. And this is one of the Nanji papers. He's got a few good ones comparing the effects of different fats in terms of alcoholic fatty liver disease that are show some pretty dramatic effects in terms of protective effects with saturated fats and extremely negative effects with unsaturated fats, especially fish oil. So the title of this paper is Dietary Saturated Fatty Acids, Reverse Inflammation and Fibrotic Changes in Rat Liver Despite Continued Ethanol Administration. And again, this is one of those Nanji papers. And the quote states, "We investigated the potential of dietary saturated fatty acids to reverse alcoholic liver injury, despite continued administration of alcohol." So they're going to cause a alcoholic liver injury, continue feeding ethanol or alcohol, and then feed different fats. Rats in group 1 and 2 were fed a fish oil ethanol diet for 8 and 6 weeks respectively. So these ones started with fish oil and alcohol and then continued with it. Rats in groups 3 and 4 were fed fish oil and ethanol for 6 weeks before being switched to isocaloric diets containing ethanol with palm oil in group 3 or medium chain triglycerides in group 4 for 2 weeks. So they were on the same initial diet, then switched for just 2 weeks to palm oil or MCTs with the alcohol instead of fish oil. And then rats in group 5 were fed fish oil and dextrose for 8 weeks as kind of a control. The most severe inflammation and fibrosis were detected in groups 1 and 2, as were the highest levels of endotoxin, lipid peroxidation, activation of NF-kappa beta, and mRNAs for COX-2 and TNF-alpha. These are the rats that were fed fish oil and alcohol the entire time. By the way, just for reference, they're using a fish oil and alcohol diet to cause fatty liver issues and alcoholic liver injury. They're intentionally, because that is the best, that creates it even more dramatically and quicker. then they're switching it after. So in groups 1 and 2 that stayed on fish oil and alcohol, they had the highest levels of endotoxin, lipid peroxidation, and all sorts of inflammatory mediators, NF-kappa beta, TNF-alpha. Then we're looking at those other groups, the groups that were just switched for 2 weeks to the other fats. So it says after the rats were switched to palm oil or MCT, there was a marked histological improvement with decreased levels of endotoxin and lipid peroxidation, absence of NF-kappa beta activation, and reduced expression of TNF-alpha and COX-2.
But they were still drinking. They were still drinking alcohol.
Right, they were still having alcohol. Yes, they were still having alcohol. And in just 2 weeks of having saturated fats in place of the fish oil, there were these major improvements. And they then conclude that a diet enriched in saturated fatty acids effectively reverses alcohol-induced necrosis, inflammation, and fibrosis despite continued alcohol consumption. The therapeutic effects of saturated fatty acids may be explained, at least in part, by reducing endotoxemia and lipid peroxidation, which in turn results in decreased activation of the inflammatory mediators, NF-kappa beta, and reduced levels of TNF-alpha and COX-2. So, there's so much meat to these papers, to these studies that Nanji did, and he's done a handful of different studies, with different iterations of a very similar protocol. But essentially, what you see is fish oil is a great way to enhance any sort of inflammation and oxidative stress. I mean, we've seen this in all the mechanistic data. We see it in the animal lifelong data. I mean, it's pretty clear, and it's very clear here. And you see a very clear opposing effects from saturated fats.
If you enjoyed that clip, you'll definitely want to download the free Energy Balance Food Guide. The Energy Balance Food Guide will help you determine exactly what to eat to optimally support your metabolism and help you lose weight, improve your digestion, get amazing sleep, boost your energy, and so much more. The food guide makes it extremely easy to get started with a bioenergetic approach to optimizing your health. So head over to jfeldmanwellness.com/guide to download your free Energy Balance Food Guide.